Hello, everyone. I hope you can hear me from the back. Okay, welcome back. Last week, we started talking about minerals as another group of micronutrients. And for our discussion last week, we focused on major minerals. And so what we're doing today is talking about the minor minerals, which are also referred to as the trace elements. And so what I'm hoping we can do today is to review our slides, have our discussion on trace elements, and then I have another set of slides that are attached on Kirkus, which focuses on some important concepts that we've talked about throughout the term. And typically they can be problem areas, so I will just brush over them one more time. And then if we have time at the end, we can just open it up for questions and answers related to whatever we have discussed in class. What I'm hoping we can do tomorrow is to do a Mentimeter session again, focused on the second half of the topics that we've talked about in class this semester. Okay. So we're just going to start off with the trace elements, or minor minerals. And so if you recall the slide, can you hear me at the back? Maybe just a thumbs up. Yeah? Okay, perfect. Okay. So you may recall this slide from last week. We talked about sodium, potassium, talked about calcium, phosphorus, and magnesium, and the role that they play specifically in bone health. And then what we're doing today is focusing on iodine, iron. There is a supporting document, I think it's SD10, on zinc, copper, and manganese, zinc, copper, and iron interactions in the body. And so if you haven't seen this one yet or taken a look at it, I would recommend you take a look at it as well. Okay. So the first trace mineral that we'll discuss is iodine. And iodine is the second most common micronutrient deficiency in the world. Iodine is typically found in the ocean, and so it makes sense that it is present in plants that are closer to the coast and seafood that come from the sea as well. So the further away from the ocean, or the higher the altitude, the less iodine one would typically find in the soil. And this also translates to how much iodine you'll find in the food that is grown in that soil. So in order to prevent iodine deficiency, iodine is typically added to salt for household use. So in Canada, all salts intended for the table is iodized. So keep in mind, this is just table salt or salt that you might use in cooking at home, and very often it's indicated on it. So food processors, for example, if we think of a food industry, the processors do not use iodized salt, which is probably a good thing because they add much salt to a lot of foods, right? And so we do not want to risk running into iodine toxicity because that will be of concern. So one of the first countries in the world to iodize their salt is Switzerland, and it makes sense if you think about the high altitudes in Switzerland, because they did have problems with iodine deficiency because of their high altitudes in the Alps. And then when you think about how far away they are from the ocean, it does make sense as well that they will have iodine deficiency issues, and therefore would want to add iodine to their salt supply. Okay. So when we look at developing settings, there are disparities in the burden of iodine deficiency. And you do see this as you move away from the coast. And so some of the highest burdens when it comes to iodine deficiency is happening in regions that are farther away from the sea. So in populations that are closer to the sea, who have access to seafood, very often you do not find very high levels of iodine deficiency. So iodine, is required in the body as a component of the thyroid hormone. And this thyroid hormone is derived from the amino acid tyrosine. So if you recall from our very early weeks, we talked about tyrosine, and it is the conditionally essential amino acid in people with PKU, phenylketonuria, right? Where they cannot metabolize phenylalanine, and so they cannot produce tyrosine, which is typically produced from phenylalanine. Okay. So back to our iodine. We did say that iodine is a component of the thyroid hormones, and there are two of these hormones. So we do have T3, which is the triiodothyronine, and then we have tyrosine, which is T4. Okay. So just wanted to point out a few things that we're seeing here, and it's shown on the slide, but just wanted to highlight that. And so we talked about thyroid hormones. These are T4 and T3. They typically do circulate in the blood, bound to a plasma protein. And so we can see that being indicated here in step 1, where you have T4 being attached to the plasma protein. Okay. So T4 and T3, keep in mind these are hormones. They enter the cell where a selenium-containing enzyme is able to convert T4 to T3. And this is very important. This is indicated here in step 2. And this conversion is very important because it is T3 which now goes into the nucleus, acting as some kind of a ligand and binds to the nuclear protein receptor. So if you recall, we've talked about, in this course, we talked about vitamin D playing a similar role. We also saw forms of vitamin A also acting as ligands and then binding to a protein, which then becomes that transcription factor. And so we see T3 playing that same role, a similar role here. Okay. But it's important to keep in mind that these three ligands, whether we're looking at vitamin A, vitamin D, or iodine, each influence different proteins and different metabolic pathways in the body. Okay. So here we have T3 acting as the ligand in the expression of, for example, RNA polymerase, which is essential for the transcription of DNA into RNA. Okay. So T3 influences the synthesis of various other proteins, and these proteins are involved in especially energy metabolism in the cell. Okay. So moving on to, let's look at step 4. You have the T3 protein receptor complex then binds to that specific region of the DNA where the gene needs to be expressed. And so we see that in step 4, where you have that complex, which is now your transcription factor, being attached to that region of interest. And so what you have now is that the DNA is copied into an mRNA, which that is the process called transcription, and then it is translated, and then the needed protein is expressed out of that process. Okay. So iodine deficiency results in reduced levels of the thyroid hormone, leading to a condition called hypothyroidism, which is characterized by a reduced metabolic rate. Very often there's fatigue, and then there's also weight gain that is as a result of hypothyroidism. So one of the major symptoms of iodine deficiency is goiter, and a very extreme case is shown in the photo on the right. So when there's a decrease in the thyroid hormone, you have the thyroid stimulating hormone being released from the brain, as shown in the figure on the left. And this results in the uptake of iodine by the thyroid gland. So in cases where you have the levels of iodine in the blood being very low, then the thyroid gland begins to enlarge because it's trying to get as much of the iodine from the blood as much as possible. And in doing this, it expands. And that is what often leads to the enlargement in the throat that you see. And so this is just as a result of enlargement of the thyroid gland. And so there will be an increase in the amount of the thyroid tissue to enhance the ability of the gland to capture as efficiently as possible any iodine that it might find. And so if the iodine deficiency is very mild, this increase in thyroid size may be sufficient to ensure adequate levels of iodine. However, if you have a very severe case of iodine deficiency, you can have hypothyroidism developing as a result of it. So this can be especially disturbing if it's in a woman, because what happens is that if the woman gets pregnant, then the fetus is also at risk of cretinism, right? And so you do have iodine deficiency presenting itself in a syndrome called cretinism. And very often with cretinism, what you're observing is developmental delays, there can be deafness, and growth failure. And so very often you have stunted children, or the fetus is stunted, and then there are a lot of developmental delays as well. Okay. And so it's important to note that cretinism is described as one of the #1 causes globally of preventable mental retardation in the world. So this is a pretty serious deficiency. So when you look at the textbook, it describes the case study of Nisi, who develops goiter because she lives in a village where they rely mostly on subsistence farming. And because subsistence farming is the norm, essentially what this means is that she's eating what is grown locally. So chances are they're eating what they grow or what someone in their community grows. Okay. And because this food is being grown in soil that is iodine depleted, the food also is depleted in iodine. And so the solution, when you look at the case study and read it a bit more, the solution for her village was to add iodized salt to their food. And so it's very common, and iodized salts are highly recommended and promoted in developing studies, especially if they live further away from the coast, to reduce some of these deficiency symptoms that we've talked about, such as goiter and cretinism in children. So in Canada, just to reiterate, because I did say it earlier, salts for tabletop use are iodized. However, it is common to find some types of salts that do not have any iodine at all, that are meant for table use. However, when it comes to salts that is used by the food industry or processors, it is not, right? And so you can find salts that are not iodized, such as the Himalayan or some other types of salts that have some naturally existing iodine in them. And so they're not necessarily fortified. So one other trace element that we'll be looking at here is chromium. Okay. And one important thing that we are highlighting here is the fact that chromium enhances insulin function by activating a peptide that brings it to the insulin receptor. So chromium is required to ensure that insulin is functioning properly. So insulin binds to a receptor on the cell membrane, and then through a process of signal transduction, it promotes the uptake of glucose by the cell. But this signal is not efficiently communicated if there is insufficient chromium in the cell. So glucose uptake by the cells is reduced when chromium is deficient, as shown in the figure's arrow size. So chromium supplements, however, are sometimes marketed to people with diabetes for this reason. But keep in mind that chromium deficiency is not the main reason that the people have diabetes, and we've talked about type 2 diabetes and how it develops in this course. So most people with diabetes, especially if we're looking at our Canadian setting, they are not chromium deficient, and you only benefit from supplements if a deficiency is present. And the reason for a low uptake of glucose is as a result of not having chromium, right? However, we do see very often that chromium is promoted to people with type 2 diabetes. Okay. So now we move on to iron. And iron is the most common micronutrient deficiency in the world because you can find it in developing settings, low resource settings. You can also find it in high resource settings as well. So as a nutrient, iron is poorly absorbed. And I guess that's why you can find it everywhere. It's pervasive. There are two forms of iron in the diet. So we do have the heme iron, and then we have the non-heme iron. So typically heme iron, which is in the form of Fe2+, the ionized form of iron, is largely from myoglobin in animal sources. So you can think of things such as muscle meats. However, when we look at non-heme iron, it is found mainly in plant sources, right? And it's not as well absorbed as heme iron. And that's why you might hear that having iron from animal source, you have a higher bioavailability of it than having it from a plant source. Okay, so one way to increase iron consumption without increasing your kilocalorie intake is perhaps to use iron cookware, which when you cook acidic foods in it, it will tend to leach iron into the food. So that's one way. And so there's an interesting study from way back, just a long time ago, where a very low resource setting in Ethiopia was studied. And typically, as one will expect, because it's a low resource setting and they don't have a lot of consumption of animal source protein, the expectation was that they will be iron deficient. However, after the survey and blood hemoglobin was taken and measured, blood was taken and hemoglobin measured, it was found out that they had really good iron status. And that was because if you know the traditional Ethiopian food, I'm blanking on what it's called, injera. So it's made from teff, and it's quite a good source of iron. And it became more bioavailable when they cook it in their iron cast pans as well. And so although they didn't have really high levels of animal source protein to give them iron, they were getting really good iron from their cookware and from the food that they were consuming. Okay. So another way, we talked about using iron cookware with acidic foods. Another way of increasing iron intake will be to consume it with vitamin C, especially if you're consuming it from a plant source. And so adding vitamin C, we did talk about the non-heme iron, which is in plants, the Fe3+, that can be converted to the heme form, which is the Fe2+, and the Fe2+ is more easily absorbed by the body. And so vitamin C is able to help with that conversion. So what are some functions of iron in the body? The function of iron in the body is quite well known, and it's an essential component of the heme group, which sits inside the proteins, hemoglobin, and it also can be found in myoglobin. So iron is needed for the binding of oxygen. So when we inhale oxygen into the lungs, it goes into the blood and binds to the iron atom in the hemoglobin. And it is the hemoglobin that is able to then carry the oxygen around and deliver the oxygen to tissues where it participates in aerobic metabolism. And we've talked about aerobic metabolism where we talk about glycolysis and it going through the Krebs cycle and going through the ATC as well. So this is just showing a structure of hemoglobin. It's made up of 4 subunits of protein, as we can see. And then we see the iron embedded in that structure. So this will be an example of a quaternary structure of protein, where you have 4 subunits coming together. Other proteins might have 2, might have 3, showing different structures as well. Okay. So here just showing the chemical structure with the iron embedded. This is looking at the heme group, and we can see the iron embedded in there. So iron is a very important nutrient, and its absorption has been well studied. So non-heme iron, and just to reiterate, because I did mention this in the previous slide, non-heme iron, which is in the ferric form, is converted to the ferrous form, which is more absorbable. And iron supplements often contain ascorbic acid, because an ascorbic acid is vitamin C, and this is because it promotes the conversion of the iron to the ferrous form. Okay. It's also known that consuming animal proteins from meat, fish, or poultry enhances absorption of iron as well. Okay. So for example, if you added like ground beef to chili, for example, you will enhance the absorption of the iron from the beans. So beans is a good source of iron, but it's in the non-heme form because it's a plant source. Okay. Or you could eat a citrus fruit after your chili for dessert, and then that will help with the absorption as well. Okay. So this is just showing how iron is absorbed in the body. Here we see the absorption of iron in both forms, in the heme and the non-heme. So once iron is in the enterocyte, the ferrous iron form of the iron is converted to the ferric form. And then you can have one of two things happening at this point. So if iron intake is high, some of the iron will be bound to a storage protein in the enterocyte called ferritin. So it's important to remember that ferritin is that storage protein for iron. And this ferritin can be eliminated through fecal matter, for example, if the enterocyte dies, right? And then it's excreted. Alternatively, you can have a copper-containing protein, which will transfer the iron from the enterocyte into the bloodstream, bound to a protein called transferrin. Okay, so the storage form is ferritin, and then in blood, it is bound to transferrin. Okay. So transferrin will transport the iron to various parts of the body, and this includes the bone marrow, where you have hemoglobin being synthesized, and then you have red blood cells also being synthesized by the bone marrow. Iron will also be stored in the liver, bound to the protein ferritin. So when we think of storage, we think ferritin. We think of its transport, we can think of transferrin. Okay. And so sometimes when you have iron present in very high amounts, the ferric forms a complex called hemosiderin. And so very often iron loss occurs mainly due to the loss of blood. And for women of reproductive age, if you recall when we reviewed the DRIs, iron recommendations for women are a bit higher because of the monthly loss of blood through the menstrual loss. So just to point out a few things here, we can see a non-heme iron shown. Very often, it's not the Fe3 structure is not being shown here. But for it to be taken into the enterocyte, it has to be converted to an Fe2+, which is the ferrous form, right? And that is how it's absorbed in. If you consume heme proteins, the type of iron that is typically gained from heme proteins from the animal source is already in the form Fe2+, right? And so it is absorbed in there. We talked about that copper-containing protein that helps with attaching the Fe2+ or your iron to the transferrin, which is the way in which it is transported in the blood as well. And so it can do a number of, can take one of these three pathways, whether it's going into the bone marrow to synthesize heme and red blood cells, hemoglobin, or it can go into the body cells. It can be stored in muscle as well, or it can also be stored as ferritin in the liver. Okay. And then when we have really high levels, you have it going into hemosiderin, also another storage form when you have very high levels of iron available. So it's important to note that iron is always bound to a protein to avoid oxidative stress, because iron can play a role in what is called the Fenton reaction, leading to the production of oxidative species, right? So when we think of ROS, it can contribute to formation of ROS, where you have a lot of peroxides being produced. Okay. So the transport and storage of iron always sees iron bound to a protein. And this is to avoid, as we said, oxidative stress. So as shown here, just paying a little attention to the Fenton reaction, we can see iron reacting with hydrogen peroxide here, right? And then it leads to production of these OH groups, which perpetuates the reaction, leading to production of even more peroxides. Okay. So this reaction can cause damage to the cell because these are oxidative species that are available. So transferrin is a protein to which iron is bound when it moves through the blood. And then once it gets to the cell, it binds to transferrin receptors. So there are receptors on the cells that recognize transferrin and its attachment to iron. And so once it recognizes it through the receptors, the iron is taken into the cell. One important thing to remember here is that you have an increase in expression of these receptors. So transferrin receptors increase in number when the cell needs more iron. Just wanted to repeat that. You have an increase in how many transferrin receptors are available when the body or the cell needs more iron. Okay. So excess iron not used in metabolic reactions is, as we said, is stored with ferritin or as hemosiderin. And just to reiterate that iron losses are very often as a result of losses through some form of bleeding. So here you can see a comparison of the recommended daily allowance, the RDA for adults and adolescents. So among men, when you look at men, on your left, you can see that the requirement at 8 milligrams per day, and it's a bit higher in adolescents because during adolescence, boys are synthesizing a lot of muscle as they go through their growth spurt and a lot of myoglobin, right? The requirement for iron, when we look at women, is 18 milligrams per day, especially when you're looking at adult women of reproductive age. And this is to compensate for the monthly losses due to the menstrual cycle. When you look at adolescent girls, they have a slightly lower requirement, although it's higher than that for their male peers. Because iron, and this difference is because iron from plant sources is not as readily absorbed as iron from animal sources. And so individuals that are getting iron solely from vegetarian sources, we can see that the recommendation is even much higher if you're getting your iron mainly from vegetarian sources. Okay. And so here we can see for men, it goes as high as 14 milligrams per day, again, to compensate for the non-heme form in which it exists. And then for vegetarian women, it's 32 from 19 to 50. This is the reproductive age group to compensate for having a lot of non-heme from plant sources. So what happens when iron is deficient? When iron is deficient, we have iron deficiency anemia, which is characterized by reduced capacity of the blood to carry oxygen, because if you recall, iron is important for formation of hemoglobin, which is responsible for carrying oxygen. And so typically when hemoglobin levels are low as a result of insufficient iron, then the capacity for the blood to carry oxygen is reduced. And so typically you will find symptoms of or people who are deficient with, as a result of iron deficiency, having breathlessness, extreme fatigue, lack of energy, and this is often due to the lack of oxygen moving around to produce the energy that they need. Remember oxygen's role in aerobic respiration and in release of ATPs as well. Okay. So as shown in this figure, we can see that very often iron deficiency anemia is leading to microcytic, where microcytic refers to smaller sizes. So this is a bit different from when we looked at folate, where you have macrocytic cells, so bigger cells. But here we have smaller cells, and these cells are paler, so they are hypochromic, they are lighter in color, right? And so they are not as you would expect the red color of the heme, they are not as red as they need to be. Okay? And this is a sign of iron deficiency anemia. And as we said earlier, the physical symptoms will include fatigue and lack of energy. So it's important to keep in mind that anemia develops gradually. Okay? So here we see there are 3 compartments for iron in the body. We talked about the storage form, the transferrin, which is like the transport form, and then the third part that we see is iron that is being stored in RBCs, or red blood cells. Okay. So iron stores in the liver typically bound to ferritin, and ferritin that circulates in the blood originates from ferritin in the cell, and it's always in equilibrium with protein in the cell. So when we measure serum ferritin levels, it's an indicator of the level of iron that is stored in the body. And so, for example, if you went to the doctors with fatigue or not having enough energy, and they were suspecting iron deficiency, very often if the physician wanted to have more information about it, they wouldn't just do a hemoglobin level or hematocrit. Hematocrit is hardly used these days, but at least they would do a hemoglobin level to understand how much iron you have stored in your red blood cells. But in addition to that, what is more informative is also checking your ferritin levels to know how much iron stores you have in your body overall, right? Because by the time you have a drop in your hemoglobin levels, that is an advanced form of anemia, right? So very often, this is how it starts. When dietary intake is insufficient, iron stores are depleted. And so very often, you will see your serum ferritin levels begin to drop first. So as this continues, the amount of iron that is bound in the transferrin also declines as you have less and less iron being transferred from the enterocyte to transferrin. So despite this, decline of iron in red blood cells may continue to remain normal. So it is possible that you have a drop in your ferritin levels. However, your RBC levels of hemoglobin look normal, right? And that's why it's important that very often tests are taken for RBCs, but also it's important to understand what is happening with your ferritin levels as well. Okay. And so eventually, as the diet becomes continually insufficient in iron, and there's less and less biosynthesis of hemoglobin, you will have now changes in hemoglobin, where you will see a drop in hemoglobin levels. If this continues, then you will start observing the microcytic cells, the smaller cells, and also the paler cells that are as a result of iron deficiency. So in this slide, we see the progression from normal iron status to deficiency. Okay. So here we're starting from normal iron status, adequate iron status, where your storage is fine, the transferrin levels are fine, and then iron in RBCs are also fine. As it progresses, as the depletion starts, end progresses, you will have a drop in your storage levels first, and that will be followed by a drop in how much iron is bound to your transferrin, and then eventually you will have lower levels of circulating iron in your RBCs as well, your red blood cells as well. Okay. So I just wanted us to take the next 2 minutes to think about the reverse process. What changes occur when you move from deficiency back to the normal state? What do we think happened? So let's say you are here at this point, and then now you start being replenished with iron. What do we think will be observed? Maybe let's give 2 minutes for us to talk to someone next to us, because I've been talking for the past 30 minutes. What do we think? Okay, so anyone wants to share what they think happens? Yes, right. Yes. Right. Right. Okay. So here we will see the reverse happening. And so just because you see your hemoglobin levels come up to normal levels or increasing does not mean that one is in the clear. Ideally, you might want to see your ferritin levels also restored so that you know that you are in the right place. So very often you will be given iron supplements if you're iron deficient, not just for a short period of time. They will ask maybe for 3 months, they might check your hemoglobin and check your ferritin again. And depending on what the physician wants to see, ideally having restored ferritin levels in the right ranges as well. What was the question? Yes. Why do they measure it in serum? Because I would think of it as the ease of it being measured, right? If you're looking for a biomarker, I think the blood is the easiest way to do that instead of taking a bit of the muscle. I don't think people will be very happy about that. Because yes, it does. So that will be like a best proxy measure for how much ferritin you have in your body. Because ideally, if the tissues are saturated and they're in the right levels, that will be reflected in the blood serum ferritin levels as well. And so typically that will be measured. I mean, another way, if you were looking at mice, perhaps you could check their muscle to see, but not humans. Right? Yeah. Yeah. So moving on with iron, although the functions of iron in the proteins hemoglobin and myoglobin are well known, iron is present in many other proteins and enzymes as well. So one important enzyme is catalase, And catalase, what it does is that it's able to reduce oxidative stress by converting hydrogen peroxide to water. And very often it requires iron as a cofactor, helping this enzyme catalase to perform that function. So we will just take a minute to talk about the nutritional needs of pregnancy, because we do know that it's a life stage. When it comes to this life stage, iron status is very important, because when there's iron deficiency during pregnancy, it very often leads to very negative birth outcomes. So pregnant women require a lot of iron. The RDA is 27 milligrams, so that's even much higher than women of reproductive age who are not pregnant. And this is the amount that we typically will see in prenatal supplements, such as the one that's shown here. So the reason that iron requirements are so high during pregnancy is that the newborn infant is born with adequate iron stores. If the mother is optimally nourished and has good iron status, the fetus that is born should have good iron stores that will last them for the first 6 months. And so breast milk is a good nutrition, the best nutrition for the child, but the iron levels are not as high because we're relying on these iron stores for the first 6 months of life. Okay. And keep in mind that this iron is being taken from the mother's body, and it has to be replaced to avoid the mother developing iron deficiency anemia. So after 6 months, it's assumed that the infant will begin to eat solid foods, and these solid foods will, ideally, complementary foods or weaning foods should have good iron in them to help with their growth. And so as we said, breast milk does not contain a lot of iron. On the other hand, it contains calcium, which is required to build infant bones. One thing to remember from here as well is that calcium and iron often compete for absorption in the body. So in this prenatal vitamin, the amount of calcium that has been added is relatively modest. It is only about 25% of the RDA for calcium. And this is why it is also recommended that when you're taking iron supplements to improve your iron status, that you do not take it with milk. And you try to have milk products at different times from when you're taking your iron supplement, because they compete for absorption. Okay. So this is just a quote that I wanted to share. Iron is the #1 micronutrient deficiency in the world. It significantly affects women and children, as we can see from this quotation from a United Nations report on micronutrient deficiency indicates. Note how it refers to the nagging tiredness, right? And so because of that lack of energy that is as a result of iron deficiency, each task becomes extremely difficult. It's an unwelcome effort. And very often in children, we know that iron deficiency anemia affects their cognitive development as well. And so children who are iron deficient may not do as well in school, not because they cannot learn, but just because physiologically they are unable to do so. And there's a cognitive decline that happens with iron deficiency anemia. So one other thing that I wanted to point out here is that because very often with childbirth, there is some loss of blood, which is unavoidable during childbirth. Iron deficiency has been linked to about 50,000 deaths per year during childbirth. And the high, especially in countries where there's high maternal mortality, iron deficiency is one of the contributors to these high levels of deficiency. Okay. So with iron deficiency anemia, a woman simply doesn't have the iron stores to compensate for this blood loss, and very often it leads to fatal outcomes for mothers as well during childbirth. Okay, so this is another critical thinking exercise or activity that you will find in your textbook. And this illustrates the interaction between calcium and iron. And so we talked about how calcium and iron very often are competing for absorption. And so in this case study, we have, Jamar, 8-year-old. When you look at the diet, he does consume meats and grains. So from the meats, we do know that he will get some iron from, because it's an animal source food, high in heme type of iron, which is absorbed quite readily. However, he drinks an excessive amount of milk. So for an 8-year-old, the recommendation for milk is to keep it to 2 8-ounces a day. And so when we talk about excessive amount of milk, chances are he's going way over the 2 8-ounces of milk. He's also physically inactive, gaining weight, and it was found that he has iron deficiency anemia. Okay. And so from the case study, when you read it, it was assessed that the iron deficiency anemia is as a result of the excessive consumption of milk, because there's so much calcium that it's able to get absorbed much more than the iron that is in the food. And in addition to that, because he's physically inactive, we know that iron deficiency anemia gives us that drop in energy, and so he's not able to engage in as much activity because he's tired all the time. And this contributed to the weight gain and resultant overweight or obesity that we found in this case. Okay. And so this shows us also that iron deficiency anemia is not a standalone problem, but then it can contribute to other conditions. And so when you look at the whole spectrum of malnutrition, we can see iron deficiency anemia perhaps being more undernutrition, being undernourished in something, but we see how that feeds into overnutrition as well, where you're gaining weight, being overweight, and developing obesity. So we've talked about the consequences of too little iron. But again, if you cast your mind back to our U-shaped curve, where we said enough is good, but not more, the U-shaped curve that we've been talking about when we're looking at DRIs, we've seen often in our discussion of micronutrients that when too much of a nutrient is taken in, that could also lead to toxicity and lead to other problems as well. Okay. So iron toxicity can result from misuse of iron supplements, and very often when there's a case of iron toxicity, it's coming from supplement use. And it's typically found in children. And children are most at risk because they're able to get their hands on a supplement and take too many pills. And so for the cases of toxicity that are found, very often it's as a result of a child getting into a supplement that he's not supposed to be taking. So the reason that this overdose is so dangerous is that instead of having iron bound to proteins in the blood, now you have so much iron that you have free ions moving around. And if you recall our discussion about the Fenton reaction, we see how iron-free ions, I-O-N, right? Free ions can perpetuate this Fenton reaction, leading to overproduction of these reactive species or oxidative species as well, and then doing damage to whatever biological molecule is available. So the treatment for this kind of iron overdose is to add what is called a chelating agent into the blood. And so this chelating agent binds the iron and makes it inactive. So eventually the chelated iron is excreted in the urine as well. Okay. However, important to note here that immediate action is essential to prevent serious harm or death as a result of having free ions moving in the body. Okay. So the disease hemochromatosis is a consequence of iron accumulation, and it leads to tissue damage, typically treated by frequent blood donations and chelating agent. So key thing is, if you have a child overdose on supplements they weren't supposed to get in, that they see a doctor immediately to prevent the long-lasting damage that can occur. So I did talk about supporting document 10 earlier, and supporting document 10 looks at zinc, copper, and iron interactions. And it describes two additional trace minerals that we haven't talked about yet. So it talks about zinc and copper, and then it briefly mentions manganese as well. One thing to remember, important things to remember from this supporting document is that zinc plays an important role in maintaining immune function. It also plays an important role in growth as well. And so the supporting document also talks about anti-nutritive factors, which are very often found in plant foods. And so these anti-nutritive factors such as phytates, tannins, they do bind onto the zincs and other minerals as well, making them less bioavailable. And so there is a case study, I think we'll look at it in the next slide, related to that. Another thing to remember from this supporting document is that there is an important interaction between zinc, copper, and iron. And so when you have an excess intake of zinc, it interferes with absorption of copper. And because copper is required for the absorption of iron, it reduces iron absorption as well. Okay. So in the extreme case, this can lead to anemia as well. When you have very high levels of zinc intake, that could lead to anemia because zinc competes with copper, and copper plays a role in absorption of iron. So before we go to this, there is either in the supporting document or in your textbook, there is another case study where it talks about a child living in a low-resource setting, mainly consuming a diet of breads made from whole grains, but does not have very high intake of animal source foods as well. And the question was, the child shows up stunted, meaning they're too short for their age, and this is not as a result of natural variation, as a result of malnutrition. And so the question was, what do you suspect to be the deficiency in this case? And very often it will be zinc, because zinc, as I said, zinc plays a role in growth overall, and it also plays a role in immune function. Okay, so keep in mind that we have self-assessment questions on Kirkus related to the trace minerals. And before we move on to our review, maybe we can take a 5-minute break. When we come back, we'll do this in-class reading activity and then move on to talk about our reviewing some concepts from the term. Yes.
There's one slide.
Hopefully.
There was one slide that I didn't get all the details
Okay.
from.
Okay, so I think we can get started again. It's been 5 minutes since we stopped. So what we want to do next is to use the link to access some information about iron. And then as we read this resource, we want to note the following things. What are some important food sources of iron? How does iron supplements contribute? What are the detriments of taking iron supplements versus having animal source foods? And then, most importantly, we want to look at groups that are at risk of iron deficiency anemia from this reading. Please let me know if you can access the link at all. And then finally, what is anemia of chronic diseases? So maybe let's give ourselves 7 minutes to do the reading, and then we can come back and answer the questions together. maybe 2 more minutes and then we can get back. Okay, so maybe we can start from some important food sources of iron that we found.
Meat, seafood, poultry, anything. Meat.
Okay, so animal source foods, specifically protein foods. Any any other sources, plant sources maybe? Yes. Spinach, yes. So green leafy vegetables in general, yeah. Any, yes.
Also like
Okay,
breads and cereals and breads are also fortified with iron.
so cereals, foods that are fortified with iron. Okay. Any, yes. Sorry, say that a bit louder. Dark chocolate? Okay. Any others? Hopefully, we're not eating huge amounts of that to get our iron status up. Any others? Yes. Legumes? Lentils, yeah. So lentils and other legumes in general, those are good sources. So which ones will be, so we've seen animal source foods versus plant source foods. So which ones are good sources of heme iron and which ones are sources of non-heme? Yes.
Animal sources are probably good sources of heme, and plant sources are typically non-heme.
Okay. So animal sources are good sources of heme iron, and then the plant sources will be good sources of non-heme. And so we also talked about how you can convert non-heme to heme, which is more readily absorbed by eating with vitamin C or ascorbic acid. Okay. So what did we learn about iron supplements? Any thoughts about iron supplements? Perhaps something interesting. Yes.
There are two amounts that are listed, and for iron, there's like the supplemental form, and then there's like the elemental form, and then the iron that's like bound to the compound. But the one to kind of pay attention to, I guess you could say, is the one that's an elemental form, because that's what's actually going to be absorbed by the body.
Any others? Any other findings from our readings about supplements? Something that you thought was interesting? Yes.
With vitamin C, it will increase absorption of iron.
Yes. So you can, so vitamin C works if it's coming from a food source or it's coming from a supplement. Any others? So we also talked about how, because calcium, so this is not from your reading necessarily, we talked about how calcium and iron compete for absorption. And so ideally, when you're taking supplements, you do not want to have it with something with dairy, which is a good source of calcium as well. Okay. Groups that are at risk of iron deficiency anemia, population groups, yes. Pregnant women, and then women of reproductive age because of the monthly loss, they are at higher risk of iron deficiency anemia. Any other groups? Yes.
Vegetarians or vegans.
Right, vegetarians. So if you recall, when we looked at the RDAs, they were much higher for people who are following a vegetarian diet, just because most of their iron sources are non-heme, and so the higher amounts is to compensate for that, to improve absorption. Any others? Any other group that's at risk? Yes. Say that again. Endurance athletes. Did it say why they're at risk?
It says running can cause trace amounts of GI bleeding and a condition called foot strike hemolysis that breaks down red blood cells at a faster rate.
Okay. So they're at risk because of GI bleeding. So we talked about how iron loss is typically through some form of bleeding, right? And so any condition, so people with ulcers might also, stomach ulcers might also be at risk of iron deficiency anemia just because they might have bleeding lesions in their gut. Any other groups linked to hereditary? Yes. Okay, so in the elderly, she said, as a result of impaired absorption that is coming from inflammatory bowel diseases, or just sometimes aging, absorption is not optimal as well in the elderly. And then typically they're at risk of malnutrition, undernutrition, which can also increase their risk of iron deficiency anemia. Did we see anything about anemia linked to some chronic diseases? Yes. Okay, so people with chronic kidney failure. Any others? So sickle cell disease, it is a hereditary disease where it affects the shape of red blood cells. So typically red blood cells are concave shaped. I don't know if this is specifically in your reading. They are concave shaped, but in someone with the hereditary disease, sickle cell, they have sickle cell shaped red blood cells, and they do not perform optimally. Very often, hemoglobin levels are low because of this change think of change of DNA, which expresses this type of red blood cells as well. Okay, so that will be one. In low resource developing settings as well, anemia can also be caused by helminth infections, so worms, right? So you can have severe cases of worm that could lead to low iron deficiency. Iron deficiency anemia as well. Okay. All right. Thank you for the multiple answers. Usually it's just a few people. And then we want to move on to just a quick review before we end the class. So we're going to use tomorrow, 1 hour tomorrow, to just go through. If you have questions about anything we've talked about, that will be the time to ask those questions. I'm going to start with some Mentimeter questions, maybe for the first 30 minutes, and then I'm going to give an opportunity for people to ask questions as well. If you don't have any questions, then we'll just do more Mentimeter questions as well. Okay. So just a reminder that our final exam is on April 27th. Believe me, I have complained about the 7 p.m. to 9 p.m. for the past couple of years, but they always put me there. And so, yeah, it's 7 p.m. to 9 p.m. I don't think anyone is listening to me about that one. Okay. And so it's in the exam center, rooms 100 and 200. The exam will be 50 multiple choice questions, similar to the type of questions that you saw on the midterm. And it's cumulative 30% from the earlier weeks before, and then 70 from the last half that we've done. So this is one concept that, based on the statistics from the midterm and other previous years, it's a concept that some may find still challenging. So I just wanted to go over it briefly, and if you have questions related to that, we can listen to that. So again, it's about the requirement distribution and the intake distribution. So one thing we want to remember is that when we're talking about a requirement distribution, if you recall, this is based on a depletion repletion curve experiment that was done. And so essentially the EAR is set at 50% at the mean or the median. And then the RDA is calculated as 2 standard deviations above that median, right? And this is supposed to meet the need. As we can see that in the tail here, we have an area under the curve of 2.5%. So essentially when someone has the RDA amount, what it's saying is that they have about 97.5% of meeting their needs. And we typically will round it off to 98%. And so when you look at the definition, you will see 98%, right? And so this is showing some areas under the curve, 1 standard deviation, what is the area there that is covered. And this is just a typical normal distribution curve, typical Gaussian curve. What happens with that? Okay. So from the requirement distribution, the EAR and the RDA can be determined. And so when you look at the back of your textbook, looking at those numbers for different nutrients, if there's an EAR, it's based on a requirement distribution, and then the RDA is set up for that. There are cases where there's insufficient information to do a depletion, repletion, or it's just unethical to do that. In those cases, you will have an AI, which is the adequate intake, which is established in place of the EAR. Okay. So how can you identify the EAR and what does it represent? So the EAR is usually that 50%. That will be the intake, and it could have a unit of gram, milligram, microgram. That will be the intake at which 50%, you have a 50% probability of meeting your needs. That's the EAR. And then what about RDA? We talked about it. That's 98% or 97.5 to be more accurate. So how is the RDA used? RDAs can be used for planning meals for individuals. Very often when we're looking at population level, the EAR is what is used to estimate how well they're doing with regards to their diet. So how about an intake distribution? We just saw what a requirement distribution is. When we're looking at an intake distribution, this is the intake of a population. And I gave an example in class of, for example, we could come to Peel and we say we want to understand the vitamin A intake of women of reproductive age in Peel. So that is the intake of a population, right? And so how do we determine the prevalence of adequate intake in a population or group? If you recall, we talked about the EAR cut point method. And so ideally, you will have an intake distribution from some population, and then you will take an EAR, which was determined from a requirement distribution curve, and plot it on this curve. Okay. And so if we're looking at the EAR, we're looking at the percentage of people falling under the EAR to establish whether it's adequate or inadequate. And so maybe I'm just going to throw it out. What do we look out for to establish adequacy or inadequacy? Is it here? Okay, it doesn't necessarily state it. So what do we look out for? What percentage are we looking at? What cutoff are we looking at? Yes, go ahead. Right. So if it's greater than 10%, we'll be concerned, right? Because even like you have a high proportion of the population not meeting even the EAR, which is set at 50% probability of meeting your needs, right? But then if it's under 10, just rule of thumb, we'll say, oh, it's okay. They're not doing too bad. Okay? So one other thing I wanted to also review is the B vitamins. So if you recall, when we're looking at we're looking at micronutrients, specifically vitamins. We looked at the B vitamins, B6, B12, folate, and we looked at the homocysteine hypothesis. This was a guest speaker coming to help us with that. And so just to point out a few things here, we see how vitamin B helps with lowering homocysteine levels by converting it from homocysteine to cysteine. And why do we want homocysteine levels to be low? Because high levels are often synonymous with increased risk of cardiovascular diseases. And so we want to keep it low as much as possible. And so vitamin B6 plays an important role in that as well. We also see how, by the help of vitamin B12, there is conversion of homocysteine to methionine the other way. And it's methionine also with the help of vitamin B12. It's not shown here, but it can be converted to methionine derivatives, right? And these methionine derivatives play a role in formation of the myelin sheet on our neurons to make sure that impulses are sent optimally around. Okay. We can also see the role when we look at number 4. We can see the role of vitamin B6 in converting folate to methylfolate, and it is methylfolate that now helps with the formation of methyl B12, also playing a role in reducing homocysteine levels by converting it to methionine. Okay, and so we see how these B vitamins play a role in regulating homocysteine levels as well. Okay, so now just focusing on folate for a second, we talked about how folate plays a role in DNA synthesis, and an example is given of its role in red blood cell formation, right? And so we do have red blood cell precursors in the bone marrow. When folate is adequate, you have normal cell division because DNA is replicated optimally. And once that is done, you have formation of normal-shaped, normal-colored red blood cells. In the event that folate is deficient, what happens is that you do not have the cells dividing optimally, and then you end up with really huge blood cells, which we are calling megaloblasts. So megaloblasts, they're cells, they still have the nucleus in there. However, when we're looking at macrocytes, they do have those big cells, but the nucleus is missing. And this is what we are referring to as megaloblastic anemia. Okay. Please let me know if you have a question related to this as I go through. Okay. Okay. One other important role of folate, so we've seen a number of roles of folate, how it helps to reduce homocysteine levels, it contributes to that. We've seen how folate plays a role in red blood cells formation, making sure that we do not have anemia, megaloblastic anemia. Another important role of folate or folic acid will be its role in neural tube development, right? And so we talked about how the neural tube typically closes in the first month after the zygote is formed, where there's fusion of the sperm and the egg. And so having low folate intake will increase one's risk of neural tube defect because you do need the folate to help with the closure, right? It plays a role in the closure. And very often this can lead, when you have low levels of folate, it can lead to what is called spina bifida, which is incomplete closure of the spinal cord or the vertebrae. And then we also have what is called anencephaly, where you have a missing brain, so you can have a skull with no brain in it, or the skull development is also impaired as well. Okay. So just to reiterate what we learned about how folate plays a role. Folate plays a role in closing the neural tube defect by its role in one-carbon metabolism, again, in DNA synthesis and methylation. Those are the three ways in which it contributes to the closure of these cells. So we can see here by day 28 of the fetus's life, the neural tube closure is complete. And so this is why the recommendation is that women of reproductive age typically have optimal folic acid status by consuming 400 milligrams of folic acid to make sure that if they get pregnant unplanned, at least they do have adequate folic levels and will not have the issue of having fetuses with a neural tube defect. Okay. So the impacts of folic acid fortification of a lot of staples, this has resulted in a reduction in neural tube defects by 50% in Canada. We also, when we're talking about folate and how it has helped with neural tube defects, we also saw the other effect where there was an increase in cancer cases as a result of, well, not as a result of, but soon after fortification was implemented. And we explained it as being as a result of exposure of pre-existing cancerous cells to a lot of folate, leading to that increase in occurrences of or incidences of cancer that was observed. When we looked at that same graph, when we're going through this part of the lecture, we saw a dip soon after bringing it back to normal levels of cancer cases after the public health recommendation for folic acid supplementation has been maintained for a while. Okay. So we've already talked about this. Just wanted to provide, more detail for your revision when you're looking at this. And in this graph, what is different from what we saw earlier is it shows the methionine being converted into a methionine derivative, and through DNA methylation plays a role in neural tube defect. It is also a methionine derivative that is responsible for formation of the myelin sheath, which is on neurons, and vitamin B12 plays a role in that, a role in the conversion of methionine to the methionine derivative. Okay. So one other question that very often, or concept that often might not be clear to students will be the issue of pernicious anemia and how that is different from vitamin B12 deficiency. Okay. So it's important to recall that pernicious anemia is an autoimmune disease that destroys stomach cells that make intrinsic factor, right? And remember that intrinsic factor is important for the absorption of vitamin B12. And so if I go back to this diagram here with when we talked about absorption of vitamin B12, we did say that vitamin B12 in food is bound to a protein. And when the food is consumed in the stomach, you have HCl and pepsin helping to release vitamin B12 from the food protein. So now the vitamin B12 that has been released from the food protein now gets attached to the intrinsic factor, which is released by the stomach lining as well. And it is this intrinsic factor, vitamin B12 complex, that ends up being absorbed into the body. And so when the intrinsic factor is not there, that's a problem because your vitamin B12 is not being absorbed optimally. Okay. And so pernicious anemia, because it attacks, it destroys stomach cells that make the intrinsic factor, it can lead to severe vitamin B12 deficiency. Okay. because B12 is not being absorbed. And the symptoms of this pernicious anemia look very much like the megaloblastic anemia, right? Okay. And so important to remember that this type of anemia, whether you're looking at pernicious anemia or you're looking at megaloblastic anemia, it does not improve with iron supplementation because that's not the cause of it, right? What responds to iron supplementation will be your microcytic hypochromic anemia, right? Where you have smaller cells that are paler, right? Okay. So here, just again pointing out, and this is a slide that you have under vitamin B12, that the black bars just indicate where vitamin B12 deficiency blocks certain pathways. And so we did see that conversion to the methylated folate, methylated B12. And so we can see vitamin B12 in its absence, this is not happening optimally. So everywhere you see a black bar, you cannot have a methionine derivative converting to myelin sheet because you do not have enough vitamin B12. Okay. And so the anemia that happens as a result of vitamin B12 is an earlier warning sign of deficiency. And when it's left undiagnosed, that is when you run into serious neurological damage. And so if you recall, we looked at that case study of Mr. Chen, where Mr. Chen had tingling and some neurological problems as a result of having some form of gastritis that affected his production of the intrinsic factor. Okay. So undiagnosed B12, as we said, can lead to neurological damage. And most folate supplements in Canada also contain vitamin B12, so that you do not have a case where you have so much folate, it is masking your vitamin B12 deficiency. Okay. Important to remember, high folate intake can mask vitamin B12, and it can lead to irreversible damage if you do not catch it early, and the neurological damage is far advanced. Almost there. So now jumping to vitamin A. So vitamin A is a fat-soluble vitamin. We talked about different forms of vitamin A. So we have the retinol, which is the active form. We have the retinal, which is involved in eye health. And then we have the retinoic, which acts as the transcription factor. Okay. So here we are looking at the vitamin A visual cycle and how deficiency of vitamin A leads to night blindness. So vitamin A deficiency can also, as we talked about, it can also lead to changes in how epithelial cells, the precursor cells, are differentiating, right? And so we talked about night blindness being one symptom of vitamin A deficiency. We also talked about xerophthalmia being an advanced form of deficiency where you see a whitish layer on the eye, and very often it leads to blindness. So what is happening with xerophthalmia, which is the advanced form of vitamin A deficiency, is that you have, because the vitamin A is deficient, it affects how precursor epithelial cells are differentiating, right? And so now you have your precursor cells, instead of differentiating into mucus-secreting cells, what is happening is that they are differentiating into keratin-containing cells. And so keratin is like what we see on our nails, like keratin is in our nails, and that is responsible for the whitish layer that you see with xerophthalmia. Okay. Here we see the visual cycle. Vitamin A is involved in the perception of light, and the form of vitamin A that is involved in eye health is the retinal. Vitamin A deficiency results in night blindness. So if we go through, just trying to go through the steps, when the eye, maybe starting from one, when light strikes on the visual pigment, rhodopsin, and keep in mind that rhodopsin is made up of this opsin structure and retinal, right? And so when you have light exposed to it, you have rhodopsin, which is formed by this combination of retinal and opsin. And then in step 2, what we see is that the retinal molecule changes from a cis shape, and a cis shape has a kink in it, so it's bent, right? To a straight transconfiguration, right? And this is what is able to send a signal so that we can perceive what is being seen. Okay. So a nerve signal is sent to the brain telling us that there's light, and retinal is released from the opsin. So in step 4, some retinal is lost from the cycle. However, if you have good vitamin A status, your retinal that you need to form your rhodopsin is readily supplied from the blood, right? However, if you do not have adequate levels of vitamin A in the blood, then there's a delay in what you see, or you may not be able to see very well in dark spaces as well. And so what we see in 5, some retinal returns to its original cis form and is able to bind, but you also need some to be replenished from the blood supply. Okay. And this is only achieved when you have a healthy vitamin A status. Okay. And so when you have low vitamin A, as a result of vitamin A deficiency, that readily or optimal conversion or formation of rhodopsin is not happening. And that's why you might not be able to see at night. Another question that I think it's a trick question that always students may have issues with is when it comes to types of amino acid. It's pretty straightforward what essential amino acids are, also known as indispensable amino acids, and also clear what non-essential ones are, right? But I think very often the conditionally essential question tricks some students. Okay. So we talked about people with phenylketonuria, PKU, and we talked about how they are not able to metabolize phenylalanine adequately. So we know phenylalanine is an essential amino acid. We cannot produce it in the body, and so we need it from exogenous sources. And typically we have tyrosine being a non-essential amino acid. And so if you do not have PKU, you should be able to convert your phenylalanine to your tyrosine, and it is non-essential. However, for someone with PKU, tyrosine becomes conditionally essential because they cannot make it because of the PKU state, right? And so then we say tyrosine becomes conditionally essential for someone with PKU. So here again, quick explanation of protein digestion and absorption. Sorry, I don't know what happened here. Protein digestion and absorption. Okay. So we talked about how proteins are broken down into individual amino acids. They are also broken down into dipeptides and tripeptides. So if you recall, we talked about a dipeptide being made up of 2 amino acids linked with a peptide bond. And then we talked about the tripeptide being made up of 3 amino acids linked with a peptide bond, right? And so very often the breakdown or the digestion of proteins leads to formation. You will have some amino acids, you have tripeptides, and there are different receptors that will take these amino acids in, different receptors that will take in the dipeptides and the tripeptides, and they are further broken down in the enterocyte to the basic unit, which is the amino acid. Okay. So the individual amino acid is what then enters the bloodstream, and they can travel to the liver, and then they're sent to different parts of the world. And so if you recall lipoproteins, we talk about very low density lipoproteins, high density lipoproteins, intermediate density lipoproteins. These lipoproteins are essentially being packaged in the liver from these proteins that are going to the liver, and then the lipids that come in, and they're packaged that way. And so the proteins are playing a role in these lipoproteins. They form part of that structure of it. One other thing, protein deficiency that I wanted to highlight here. And so we talked about protein deficiency and how it can present itself in 3 different ways. And so protein deficiency, it can result, I think this was from a guest lecture as well, it can result in kwashiorkor. And kwashiorkor is a Ghanaian word from West Africa, which locals used to refer to diseases of the first child. They talked about it as the disease of the first child once they are weaned off of breast milk when a second child is born. They saw that very often this was happening. The symptoms of kwashiorkor was happening. So it's a local word, kwashiorkor. And marasmus, the difference between kwashiorkor and marasmus is that for kwashiorkor, the child is being given foods that are high in energy from carbohydrates and perhaps even fat. But then the diet is lacking in protein. And so very often it presents as a swollen belly. And then you can have edema or swelling in the feet and in the hands as well. And the swollen belly can be as a result of fat accumulation, because we just talked about how proteins are needed to repackage lipids in the liver. And so when there's no protein, what happens is that there's an accumulation of lipids in the liver enlarges, and then the stomach enlarges as well with that. Protein deficiency can also present as marasmus. And very often you're seeing marasmus when there's deficiency of not just protein, but energy in total. So essentially hunger, very low caloric intake, right? And so with the low caloric intake, what you often see is that there's wasted muscles. And this wasting of the muscles can show up in the face, it can show up in the gluteal region, that's the behind, and it can also show up in the arms, anywhere there are muscles, right? And so very often you see the child looking like a very old person, because they have lost all their muscle, and they don't look like a typical child. And so what happens is that when you're thinking of marasmus, you're thinking of wasted muscles. There's no edema with marasmus. But some cases you can have a child with marasmus and kwashiorkor, and that's what we're referring to as marasmic kwashiorkor. Here you still have the wasting of muscles that you do see in marasmus, but then this child also has swelling, edema as well, which is kwashiorkor, and a typical characteristic of kwashiorkor. And so that will be the difference between the 3 conditions. We're almost there. So we talked about iron absorption today. So this is just a quick review because we just saw it. And if you recall, we saw this where non-heme iron is very often, that is Fe3 plus is converted to Fe2 plus, which is more readily absorbed in the body. And once it's absorbed, you have a copper-containing protein that helps it to go into the blood attached to transferrin. And it is in this form that iron is transported in the body. So it can go to the bone marrow to form red blood cells. It can go to, for example, the muscles to make myoglobin. And then it can also go into the liver to be stored as ferritin. And so that's just a quick review. And we've had a lot of activity today talking about heme, non-heme, and the differences between the 2. Okay. So that was my final slide, and a quick review of some concepts that sometimes students may struggle with, what they hear in tutorials and the questions that I do get from office hours. What we're hoping to do tomorrow is to have just a question and answer session. If you have questions, please bring them. I'll have enough Mentimeter, but I'll take a break 30 minutes into it to see if anyone has questions. If you don't, then we'll just spend the whole time doing Mentimeter. Thank you for staying with me. I'll see you tomorrow, and then we can have our final words tomorrow. Take care. All the best. So it's a combination where it's almost like there's not enough calories and there's not protein, and sometimes it presents itself like that.
And how is that different from just regular marasmus?
So marasmus, there's wasting of muscle, but there's no edema, there's no swelling in the feet, right? So marasmic kwashiorkor is essentially a combination of the two, and edema is characteristic of kwashiorkor.
It's basically just marasmus plus edema.
Yes.
Okay.
Yep. Yep. Hi.
So do you know, like in the city Smith Hill, like they ever doing like
I know. I think they should still be doing. There's a service. I don't know which exact place, but students have told me they have gone to Sydney Smith and they got.
I thought for the meet tomorrow, I was just wondering if that was.
I don't think they have it because the exam is usually restricted, so you might not have that. But I would say you might find questions that are still phrased in ways that, yeah,
Okay, thank you.
yeah, sure. Hi, how are you? Good. How are you doing? Really good to see you.
We had a question. So,
Right.
okay, so you know the slide on, yeah,
Right.
you mentioned like that was
Right.
an example. He just we couldn't find it in the slides or in the text. Is there a way to, is it anywhere?
Sickle cell. No, so that will be the in-class reading.
Okay,
So it's not there, but it's just additional information.
so it's not gonna be on this.
It might not be there. Let's see. It's been a while since I actually read this
It's just
thing.
because he like for preparation for the exam, it's like
What is anemia, chronic disease occurs from low antibiotics, infections, cancer, kidney. So this one is really related to inflammation. Yeah, but that's just additional information that occurred to me that I.
oh okay, okay.
No, it's not gonna. I
Okay,
don't think there's gonna be a question related to that.
And
Right.
then so so some of the quizzes he was
Right.
having difficulty on. So it's like where can you find for the
It
quiz that
depends
he-
on what questions. Mostly from the slides. It should come from the slide. So when you look at it and it's asking questions about lipids, like related to lipids, then you know it's from the lip. And I did give multiple tries. I think twice or yeah, I think twice or three times
yeah,
maybe.
twice. But he kept trying, and then you're like, I can't find it, it's not here. Like some
Which
of them
one
he
is
did
it?
really not
6,
good on, like some of the quizzes.
7. But it drops your lowest 2.
Yeah, and it's
Yeah.
weird because it's like he just wants to be able to find the information so he can review for himself.
So
Who should
if
he
you
talk
can
to? Yeah.
tell me what kind of questions is being asked, I may be able
Maybe
to.
email you them.
Yes.
Okay, we'll do that.
Yeah. And then I will have an idea which week that is.
Okay. And thank you so much. And then two more questions. One is for tomorrow's review. You're not going to be able to make it because he has a wisdom teeth
Right.
removal. Like maybe, depending how you feel after, because it's in the morning. But is there any way it could be recorded?
It should be recording right now because we have the open cast. It's on Kirkus. If you go under OCCS student app,
Do you know that?
yeah, OCCS student app, you should be able to see all the recordings
Ah, okay.
since the beginning of the term.
Oh.
Yeah.
Okay. And what's
So
your
if it wasn't captured, then it's not me. Yes, it's happened in the past, but I'm still fingers crossed. Yeah.
okay?
Yeah.
Are you having any more office hours?
I will have office hours on Fridays, the virtual ones. It's on the syllabus till the last week before the final exam.
Okay, because it says you're booked. Is that the one where you're
Yeah.
booked up?
So
Okay.
if people have already booked, then
That's it.
yeah.
And then unrelated, but what's your opinion on peptides? Because it's like really popular now. People are taking like peptides. Like, have you seen that?
No, I
Like
haven't.
injections, like
Oh.
yeah, to like
Oh, like
boost-
GUTS peptides?
Yeah. Like
Is
to
it like
boost
for
certain-
weight loss?
Could be for weight loss, or to increase certain different. It could be for weight loss. Mainly people use it for weight loss.
Yeah,
Yeah.
peptides.
Have you
So is it like GLP-1 or?
Yeah, that's like the popular one. But then there's these other ones that it's very popular online right now.
Do you know what exact peptide? Like there are a number of them. Like this peptide YY.